Good afternoon, good morning, good day, depending on where you're sitting in this virtual Boston. My name is Klaus Gustafsson, and I am the Chief Commercial Officer and one of the co-founders here at Adam. I really want to thank the PEG organizers for doing and creating this fantastic conference, despite all the COVID issues that we're dealing with. And I want to thank you for taking time of your busy day to listen to me presenting the antibody pipeline we've been developing. Adam was founded back in 2003 as DNA 2.0. Some of you may remember that name. And the company is today 120 employees strong, and we're based here in beautiful San Francisco Bay Area, where the sun always shines. As you'll notice from the talk, we're very technically driven and technically focused. So trying to really invent around many of the constraints of biology. So to this day, we have, to that point, we have maybe 30 issued patents. We have published more than 60 peer-reviewed papers. And one of my favorite is we have in order of 3,000 publications by third party that used us for something, and they referenced us in their material methods section for genes, proteins, or cell lines. So that's one of my favorite reads. It's always fun to see new things coming out. So antibodies, they of course come in all shapes and sizes, and it's really impressive to see the creativity of science being directed toward making all these novel types of molecules, especially now with all the Frankenstein molecules with the IL-2s hanging off of them and all these different things. The nice thing with antibodies is that despite the diversity of sequence and structure, the core elements are the same, and the core applications are very similar. So that allows us then to build a generic platform that we can use to optimize a number of these molecules using the same underlying tools and system. So the ecosystem we built here really is trying to look at the whole process from start to finish. How do you generate the antigen? How do you generate, the subsequent hybridoma sequencing or what have you to identify your hits? And of course, the antibody production itself, and I'll get into that in more detail in the next few slides. Production, expression, purification. We have a nice platform for engineering these antibodies for novel properties that I'll touch upon with a nice case study. And then to take these antibodies and then generate stable cell lines, we built a leap-in platform that uses engineered transposon to rapidly and incredibly stable production of high-quality antibodies in the cell line per se. So this allows you then to go from virtual idea concept all the way to research cell bank as efficiently as possible when everything is integrated under one group. So the process we built is really around, on one hand, manufacturing. How do you manufacture genes? How do you manufacture proteins? How do you manufacture cell lines as efficiently as possible, as integrated as possible? And this is all robotics. It's all liquid handling stations. It's all a system that allows us then to barcode and timestamp every single sample as it goes through the process as we build these different biological entities. One thing that may not be fully appreciated is that LIMS and the database that sits underneath all this, that captures all this sequence function information. And the LIMS is really the tool that allows us then to make this process of millions of genes in parallel and thousands of proteins and hundreds of cell lines, and still retain that all that corresponding underlying information on the function and fitness for all of them, and how that they respond to various constraints such as productivity, such as binding, such as aggregation. So what's the point of having this all this computational infrastructure and production infrastructure? Well, it allows our customers to really come in and utilize that to the best possible degree to be able to go from their discovery hits into actually validated leads. So here's an example of a customer of ours that routinely provide us with 96-fold sequences that they've identified through because these are just the CDR sequence of hits from a discovery platform based on deep sequencing. As they take these sequences, now online we can automatically recode them to maximize the expression yield, in silico clone them into battle-tested scaffolds, and then synthesize them, make the oligos, make the genes, make the corresponding proteins, and then produce antibodies that in a 96-fold plate range from an order of 100 mg per mil up to say 300 mg per mil. Right? So this is not, they're not a huge amount of protein, but it's still sufficient that we can take these proteins and now measure things like binding. So we're looking at here is 1 over KD on the y-axis, and identify which ones of all these hits that actually ends up being a decent lead for subsequent developability engineering, and to be able to do that as rapidly as possible. So here we can go from, typically we go from virtual sequence to a validated lead in 4 to 5 weeks. Now, so 4 to 5 weeks may seem like fast, but as we all know, the year of COVID, fast was not fast enough. So under the constraints of COVID, we were really pushing the system to the limit. And here's an example of a case study we did with another customer, where they came to us with the requirement of getting 70 hits they've discovered in their process to fully validated leads. And 4 to 5 weeks was way too much time, and 96-fold format was much too small scale for them to be able to validate the system as they needed. So here we really optimized the process, and you can see here they came in with the sequences. We can do all the design completely automatically, of course, in 0 time. Synthesis took maybe 5 days. Large scale plasmid another 2 days or so. We can take that large scale plasmid prep, transfect that into hex cells, express another 5 to 7 days, and harvest, and then purify, and then before shipping. So here we can go from virtual sequence to shipping things out. For the fastest one took 18 days, and the slowest one took 21 days to go from start to finish. And the nice thing with this process, because it's been optimized, because it's deeply integrated, allows us then to really engineer for maximum expression yield. So despite these CDRs coming from a silico-derived screen, we could generate sequences that on average produce in order of 500 mg per liter. So here we're looking at the clone numbers on the y-axis, and the titer, the purified titer on the x-axis. And we see a good, nice distribution with an average around 500 mg per liter. So that means we can then generate the scale rapidly in order to build the antibodies required for the customers in as a fast time as possible. Now, certainly speed is important, but quality is absolutely mission critical. And generating these molecules rapidly with the correct constraints allowed us to do that. Having this platform allows us then also to navigate and build a number of different sequences simultaneously, where we can change substitutions in a systematic way using systematic variants, and then machine learning to deconvolute what is the relative contribution. So here's an example from another case study from a customer where they came, and we had a starting point, which is a commercial molecule. We were interrogating in the heavy chain. We had identified 41 residues where we can swap in either the wild type gene, the wild type amino acid, or a substitution based off of germline information. So 2 options at 41 residues, that space is 10 in order of 10 to 12. Likewise, in the light chain, we identified 2 residues, 2 options at 36 residues across the light chain, approximately 10 to the 10. So putting those together, it actually turns out to be 10 to the 23 is the total sequence space we explored. So that's, for 10 to 23 is a very large space to search for, certainly for 3, in this case, 3 fitness functions. So what you see here is now the starting point in red. We're looking at that first set of 96, where we made a systematically varied set, meaning each of those 96 antibodies had 5 substitutions 4 to 5 substitutions in each antibody. And each substitution was seen in, say, 20% of the data set. And these 70 or so substitutions really explore a sort of 70-dimensional hypercube in that space, so we can identify and make the 96 clones that best capture that sequence function space. Synthesize those, test them for binding. What you see is many are poor binders. There are a few that are good binders. And then we can build models that captures the sequence function correlation between those two, between the substitutions and activity and binding. And in this enormous space of 10 to 3, get rid of the bad substitutions, keep the good ones, move that in, and now we condense that space from 10 to the 23 into 10 to the 4. And in this 10 to the 4 space, we repeat that same cycle, get rid of the bad substitution, keep the good guys, and now we made the subsequent set of 5 for binding. So what you see in those 5, and you see here binding on the x-axis as Ka, so 1 over Kd improvement, and on the y-axis, the titer, and on the color axis, going from gray to pink, is Tm. So the best sequences we identify in this very efficient search were these 3 different solutions along what's called a Pareto front, where you look at optimize for many different fitness functions simultaneously. We identified these 3 molecules, and they all had different sequences and different attributes, but provided 3 separate solutions to the customer's problem that all fulfilled their needs. By searching this 10 to the 23 space in 3 fitness functions with less than 200 samples measured. And finally, in the last few minutes, I'll talk about our transposase leap-in platform. I know I've seen at least 2 presentations so far under PEGS, here at PEGS, where this platform has been described by other customers and colleagues. But I just wanted to give you a little bit of background on how this platform operates and what its characteristics are. So as many of you know, transposon has been around for a long time, and in principle 4 billion years, but Barber and McClintock really got a Nobel Prize for identifying and talking about the jumping genes back in 1983. So jumping genes are brilliantly nice and simple systems. And so what you have is inverted tonal repeats that they're flanking your, either in the wild type case, the transposase, the enzyme, or for us, we put in some engineered insert, so an antibody or what have you that we want to insert into the chromosome. So these sequences then are transfected as plasmid DNA together with co-transfected with an mRNA who is a transposase. So the transposase get translated and produced, and then grabs hold of the IPR arms and then inject and integrate that into the chromosome everywhere where it can find a TTA site. So of course in the CHO genome, there are thousands upon thousands of TTA sites. The transposase looks for open chromatin, and it introduces the sequence that we can get, from a handful up to 30, 40 different integration sites across the genome. Never ever have we seen the same integration site repeated twice. So it's a semi-random integration across the genome for open chromatin TTAA. Another really nice feature is that these things are single copy integration at each site, and then of course multiple integration sites across the genome. So that because when you do regular homologous DNA recombination, you typically get all these concatenators, you get truncations, you get deletions, and all these mess that produces incorrect molecules in the end. Whereas if you do this with a transposase, now you get really nice, super clean little cassettes that come in as heavy and light IPRs, and your selection marker or whatever you have on there, they come in and integrates directly into the chromosome. So that gives you, every integrant provides the correct molecule in a relative copy number expression. And the other nice thing is that in your pool, you're going to get a nice distribution of sequences. So here's an example of when we do these things, we see almost all the clones, like in this case, 62% that are top quartile. The top 25% are the absolute majority of the cells. Whereas if you do classic homologous recombination, you get just this tiny sliver of good expressors, and then you get this long, endless tail of non-producers. So this gives you that already within one plate's worth or 2 plates, you're going to find your winners, your clones that are going to be good expressors. Also, because of the payload of these transposons is basically limitless, you can put in heavy and light and regulatory elements and anything you want, or 3 orbs or 4 orbs into the same cassette. So here's an example where we made 14 separate vectors that each had heavy and light between it with the different promoters and regulatory regions, and then transfected those as 14 separate pools, and then looked at the ratio of heavy ORF1 and ORF2, and we'll see this. We'll be able to really titrate the exact ratio of the different things. Now, this makes a big difference, not too much in regular vanilla antibodies, but if you go to bispecifics, if you go to trispecifics, and all these multi-ORF entities, often what you see that the ratio of integrants, the ratio of the different subunits makes a huge difference for the quality of the system. And again, a case study here is where we worked with a customer on looking for urgent COVID relief. So really making stable cell lines as rapidly as possible. Here it took us 38 days from including gene synthesis, plasmid prep, as I talked about before, transfection, pool selection, and then pool banking. We took that pool banking directly to the customer, and they did PET batch. And what we see here, they're looking at in the order of 14 grams per liter production. This IND is pending, and this came out of their intensified fed batch process. So incredibly rapid, incredibly efficient process to generate the stable cell lines, or in this case, stable pool that can go straight into the process. So the speed is really nice in the sense that we can control really an integrated pipeline, all from virtual sequence to the RCB, and you can already you get the 2 week for the DNA production, the gene synthesis vector construction. You do the transfection, you get 4 to 7 weeks, of course, depending on the clones and on the cell lines, and the amount you need to generate. But already at this stage, you can then derive from your pool, can get derived material, and then up to grams material that you can then bring to early PD studies or tox studies. Then you do that your 6 to 9 weeks of clone ranking and cell line cloning, and we use the Solentum system for this. And from this, then you generate your research cell bank, or go on and generate your master cell bank. At this point, we generate 7 INDs as a service. We've done in the order of 30 plus projects delivered, and I think we have around 30 licensees, or actually more than 30 licensees across the globe, including 11 of the top 20 pharma, and some of them you heard present here at PEGS the last few days. So the advantage with our pipeline really is that it's a really industry-leading speed from start to finish. Integrating this pipeline allows us then to go from virtual all the way to RCB. And as a user, you can get access to cutting edge technology without actually investing in the underlying infrastructure. And of course, that allows you to instantly scale up and instantly scale down, and allow your process to move as efficiently as possible through the pipeline. The pipeline is built so you can access it either from start to finish, or you can cherry pick your different elements that you need to get access to. We support this with flexible business models. You can either outsource the technology, or you can in-license, outsource the project, or you can in-license the technology. and everything's done here in beautiful sunny California. The IP never leaves our building. So with that, I want to thank you and appreciate your time. And any questions, please let me know. Thank you.